So welcome back after a little bit of a long weekend, and today we're going to be continuing on, and I hope nearly finishing Chapter 11. Chapter 11 is about action potentials and how is it that electrical signals get sent from the soma, the cell body, all the way down the axon, and we'll pick up that story. And to get here, though, we really focused last week over ions, right? Sodium, potassium, where they are. What did we say? Sodium is found where in greater abundance, inside or outside? Sodium, outside, whereas potassium is found in greater concentration inside the cell. Major issue as you understand that. And every cell has a concentration of sodium and potassium, which is unique to that particular cell. But these numbers are kind of similar in all cells of your body. And there's something special about neurons. And the special word about neurons is that they are excitable, right? This idea that electrical signals can be sent down a neuron. So neurons are excitable, which means that they can be stimulated and cause this surge of energy, this action potential to travel down. And we're going to see how it is that sodium and potassium are keys to this. We have to appreciate that neurons also have a resting potential, right? That at rest, when a neuron is not being stimulated, that it sits at a resting membrane potential of about negative 70 millivolts. That's a number you're going to memorize, negative 70 millivolts. It'll show up in a lot of tables and a lot of charts that you're going to be seeing today. Keep in mind that in that neuron, right, on the inside there'll be lots of potassium, on the outside there will be lots of sodium, and the negative 70 is a collection of forces that keep certain ions inside or outside the cell. The cell is leaky. The cell membrane is leaky. There are leak channels. Okay, and this is kind of just a quick review. There are leak channels, sodium leak channels and potassium leak channels in the membrane of a neuron. And it turns out that this is where ions can move down their concentration gradient. That means what? Potassium would always be trying to get out, right? There's more inside than outside, so it's going to move out of the cell. And sodium would be trying to get into the cell, right? Because sodium's in higher concentration outside. It would be, quote, naturally trying to leak down its concentration to get inside. And what I told you in the fine print is that it turns out that potassium is simply leakier than sodium. So there's always potassium leaving the inside of the cell. What effect would that have on the membrane's overall charge? Potassium's leaving. Potassium is positive 1 charge. That's going to leave the inside even more negative, right? It's going to keep that negativity. And the cell also has to keep that negative 70 resting membrane potential pretty constant. And what was the major player? This image is showing us the regular leak channels, right? Sodium's trying to get in, potassium's trying to get out. But there's another major player here, and that was the sodium-potassium ATPase, the sodium-potassium pump, right? You'll hear this used in different ways. And this is an active transporter, meaning that it requires ATP energy for this thing to keep working. And what this pump does is it unequally pumps 2 potassiums back in, right, that have leaked out. So here's, on the orange, so here's a potassium leak channel, right? Potassium's just always kind of moving out. But potassium being brought back in, right? It's being brought back in by this ATPase. Sodium, right, was always trying to slowly leak in, and we're going to make sure that it gets pushed back out, okay, by this sodium-potassium ATPase. And the stoichiometry is this: for every 2 potassiums, there are 3 sodiums. It's just kind of an imbalance. And what else is that imbalance doing? If it's always kicking out 3 sodiums for every 2 potassiums that come in, we're also helping to maintain what sort of environment inside the cell?
Negative.
Negative, right? Ultimately, right, there's more positive being shoved out than positives coming in. So all of this is going to help keep the inside of the cell membrane, the cytosol, right, the inside, the intracellular fluid, will be overall more negative. And then we watch that little video. And these are great little videos. I want you to focus on these. You can watch it over and over and over and over. And we'll see another one here on this in a minute. So the resting membrane potential, definitely watch those. Remember to watch those videos within the PowerPoint. You need to go into Blackboard, pull up the PowerPoint presentation, and go to the presentation mode, right? Full screen mode of the PowerPoints. And then in that full screen mode, you can push the arrow and watch that video on your tablet or computer. You'll also see these videos. These are part of the A&P Flicks series. And the A&P Flicks are found in Mastering, under Study Area, and then you'll see a thing that says Flicks. And go there, and there'll be 2 or 3 videos on action potential. So there's a couple different ways to get there, to view these. And I know that in some of the assignments on Mastering that I've assigned, you will see these videos again and questions asked about them. So you're going to see these in a couple of different places. They are very well done. I highly recommend them to get the whole story in slow motion figured out for you. We're going to take something that's really, there's lots of pieces, lots of moving parts here. And no matter how I present this, there'll always be something that you wished you knew when you were first hearing me describe this. So I'm just going to jump into this and we'll figure this out. This difference in membrane potential is an electrochemical gradient. Those 2 words tell us what? There's a difference, a gradient, in both the electrical charge, the inside's more negative, the outside's more positive. That's an electrical gradient. And there's a chemical gradient. We know that some molecules are in higher concentration on the inside, some molecules are higher on the outside. So we refer to this whole difference as an electrochemical gradient. And this actually creates an energy. It creates an energy difference between the chemicals and the electrical current that is going across this membrane. And there's an equilibrium. Are these molecules at equilibrium? Are sodium and potassium at equilibrium? Nope. Are positive and negative charges at equilibrium? No. Instead, though, they are where they're supposed to be, right? They are in some homeostatic balance, right? In the cell, I keep giving the cell a consciousness, but the cell is desperately trying to hold on to this negative 70 because as I keep saying, and you'll hear me keep saying, without this imbalance, without this electrochemical gradient, neurons can't fire, muscles can't contract, and lots of things in your body can't occur. So we have to keep this difference going. If we look at the ion, it turns out that potassium has a negative 90. Hold on with me for a second. If I just looked at potassium, potassium has a potential of negative 90, and sodium is positive 76. So which of these 2 appears to have a greater contribution, knowing that neurons sit at negative 70?
Potassium.
Potassium seems to be kind of driving the show. Potassium is negative 90 by itself. Sodium is a positive difference. So what we see is that it really is, in the end, sodium's important, but it's potassium that ultimately helps to maintain and hold on to this gradient. So again, potassium at rest. We've already talked about how molecules would move into and out of the cell. And if the membrane were freely permeable, if all of the potassium could go to where it would want to go, then we would get that negative 90. Okay, just to understand where that negative 90 number is coming from, not one for you to necessarily memorize. But what we see here is that negative 70, I know this is small, negative 70 on the left, that's our little voltmeter. We're seeing negative 70, as it should be, if all the sodium could just simply leave out its little membrane transporter, then if it were equal on both sides, the membrane potential would change to negative 90. Okay, so we would go from negative 70 to negative 90 if potassium could move to equilibrium. Sodium, also very important. Same idea. We talked about where sodium would normally want to be at. Let me go back to that because that's an important concept that I have in words here. Where is the electrical gradient for potassium? In other words, inside the cell it's negative, isn't it? But potassium has what kind of charge?
Positive.
Positive. So would potassium be drawn into the cell, or would potassium be kind of slung out of the cell based upon charge? Do you agree that potassium wants to be inside the cell, right? Because potassium is positive and the inside's negative, like little magnetic charges. The chemical gradient, though, is out of the cell because there's a lot more potassium inside the cell. So from an electrical standpoint, potassium wants to stay in the cell. From a chemical standpoint, potassium wants to leave. This is a little bit different for sodium. Where does sodium want to go electrically? Na plus, outside the cell in higher concentrations, it would want to be drawn into the cell. So electrically, it's being drawn into the cell, and chemically, it's also being drawn into the cell, isn't it? Because chemically, there's already more sodium outside the cell than there is on the inside. If the sodium could go to where sodium would want to go, then the membrane potential would go all the way up to positive 66. In other words, all that sodium would come flooding into the cell, right? And now the inside of the cell would be positive.
Is this if there was no sodium potassium?
This is exactly if there was no sodium potassium pump. If the ions could do what they would want to do in isolation of homeostasis, you're exactly where you need to be. So this is what would happen if the cell did not have the sodium potassium pump, right? And if there was just no control over this, where would the ions go based upon their electrical attraction and on their chemical attraction? Good catch. Okay. So again, we would go from, as it says here, negative 70, and if sodium could go everywhere it wanted to go, it would end up at positive 66. Not where we want to be, right? If we want to maintain this negative electron or this negative membrane potential. Okay, so we've talked about the resting membrane potential and the significance of the sodium potassium pump to keep everything in homeostatic change. I mentioned to you that there are leak channels. Well, last time I mentioned, why is it that ions, sodium, potassium, cannot freely cross the membrane? What is it about Na plus and K plus that does not allow it to freely move across the membrane? They're very small molecules, right? They're ions. And what is it about them that disallows them to just move through the membrane?
Can't move across the hydrophobic.
The membrane is hydrophobic, right? And that means that it is non-charged. And these ions are very charged, right? You've got a positive charge on them. And so there's a repelling going on. So ions, even though they're very small, cannot just get right through the membrane because of their charge. So they have to have a channel, right? There has to be a protein channel through which they go. And there's three basic kinds of protein channels that molecules can move through, and we're going to call them gated channels. Imagine this is a tunnel, and at the beginning of the tunnel, there's a gate, okay? And when I say gated channel, three different sorts of molecules are going to trigger the gate to go open, okay? So we have I'll start at the top. We have chemically gated channels. They're going to open when a chemical comes along and pushes the button. Just think of it that way. There are also electrically gated channels, or what they're calling here voltage-gated channels. In other words, there's going to be a voltage change, a charge change, that's going to push the button for the gate to open. And there are ligand or mechanically gated channels. Some particular molecule comes along, okay? So we've got, sorry, I just misspoke again. Chemical, some particular molecule or chemical. Voltage, electrical change is going to open the gate. Thirdly, mechanical, in other words, somebody comes over and pushes it, okay? So mechanical, there's a touch. There's a physical touch that's going to push that gate open. And let's figure out where these different channels are. So chemically gated channel. Some chemical is going to come along and push the button. Another way of saying a chemical is a ligand. A ligand is a molecule that binds to a receptor. So ligand, or you'll hear ligand by some individuals. I say ligand. A ligand-gated channel. This gate will open only in the presence of a specific chemical. When we get to the synapse, by the end of today, we're going to talk about acetylcholine. Okay, acetylcholine. Can you picture acetylcholine? We know a little bit about acetylcholine. It's released by a neuron, travels across the synaptic cleft, and then we always said the acetylcholine is picked up on the postsynaptic cell. That acetylcholine is a chemical. That acetylcholine, when it is received on the cell, is going to cause the opening of a gate. Okay? So that's a ligand-gated channel. Some sort of chemical is going to open it up, and we'll see acetylcholine as a good example of that. This is what we're going to see a lot of in synapses, will be ligand-gated channels. So here's an example of that using acetylcholine. And Here's the extracellular fluid on the top, and the white on the bottom is inside the cell. The blue is the plasma membrane. We see this interesting looking molecule in the membrane. This is the gate. And what is going to trigger this gate to open, as we see over here, we see acetylcholine molecules, little red dots. The acetylcholine molecules are going to be specifically interacting with this receptor, right, and bind in that binding site. And as a result, as you see on the right, the gate's open, right? So the gate opened in response to a specific molecule. This is a chemical voltage or a ligand voltage or channel, okay? It's opening in response to a chemical. Then there are voltage-gated channels. This is going to be what happens in excitable membranes like neurons. There's going to be some opening or closing in response to an electrical change. And we're going to see a lot of this going on. There are going to be voltage-gated potassium, voltage-gated calcium, voltage-gated sodium channels. Again, they'll open and close. They'll be activated, opened, or inactivated, closed, based upon the electrical flux that's going on in the membrane. And so here we see a picture again, trying to break this down step by step. This is your resting cell, -70. In this case, we see that the membrane has changed to a -60. That voltage change has caused the gate to open. And as a result, some molecule is moving into or out of the cell, changing the membrane. So we see in this case, positively charged sodiums, for example, are coming into the cell. What's going to happen to the inside of the cell now? It's usually negative. It's going to become more positive. Another way of saying more positive is less negative. Okay? So what we see is that as these positive charges are coming in, the inside of the cell is becoming more positive, +30. Another way of saying that, and you'll hear me say it also, is that the inside of the cell is becoming less negative. So that would be a voltage-gated channel. And then finally, there are mechanically gated channels. We're going to see mechanically gated channels when you think about nerve endings related to touch, right? If I push down on your skin, you have receptors in your dermis that are going to respond to vibration, to pressure, to touch. Because they were physically touched, mechanically opened, something will open in response to that. So mechanically gated channels. So this is touch, pressure, vibration. Mostly your sense of touch in your skin respond to those kinds of channels. And it's hard to show that, but in this picture, we're trying to show you closed gate, right? And some sort of pressure is applied near the gate. As a result, the gate opens, and now ions flood in, and once again, when the pressure is removed, the gate closes. So 3 different kinds of gates. Okay? Now, in a neuron, getting back to what we're talking about here in the nervous system, there are chemically gated channels. They're going to be found on the neuron cell body and the dendrites. There are voltage-gated sodium and potassium channels. We've already seen those. Okay? Those leaky channels, those are sometimes voltage channels. And then there's voltage-gated calcium channels, and we're going to see the impact of calcium on axons as we go through this. So keep track of these different gates. What you do not see here on the neuron, you don't see a mechanical, right? There's no mechanical here. You're seeing a combination of chemical and voltage-gated channels. So here's a neuron, classic neuron. What's its morphology? I see many dendrites, one axon, so we would call this a multipolar neuron. And what do we call this area again, right in this area? Where the axon attaches to the soma, that's the axon hillock, right? And then we have this long axon. Where are these different channels in this cell? Well, when those signals are received from other neurons coming in at the dendrites, and sometimes signals can come in right to the soma, can't they? At that location, there are going to be chemically gated ion channels. Then, right here, at the axon hillock and going down the axon, there are going to be a large number of voltage-gated channels, both for sodium and potassium. And then at the very, very, very end, at the synaptic knobs, we're going to talk about voltage-gated calcium channels as we continue through this discussion. Okay, so we have different kinds of channels triggered by different molecules, charge differences, or pressure changes. So there are graded potentials. I called them local potentials last time. And then we're going to be talking about action potentials. These are two really important things to keep in mind. A local potential, there are some characteristics here that I need you to keep track of. Local or graded potentials only travel for a short distance. They cannot spread very far. Okay, that's going to be a big distinction in a moment when I talk about action potentials. And in the neuron, when the signal comes in, here's my soma, and here are my little dendrites. When a signal comes in to the soma, that initial signal that comes in is a graded potential or a local potential. What we're going to see is that these graded potentials might have an impact and cause an action potential to go down the axon. Okay, so local potentials are going to be happening through the dendrites. Local potentials will be happening through the soma. But when we talk about the axon, we put all this together in a few minutes, we'll be talking about action potentials. And one of the distinctions about action potentials is that they can go a long way. So local potentials can't go very far. Action potentials will necessarily go down the entire length of the axon in an all or none type phenomenon. So what causes these graded potentials? We're going to start talking about sodium and potassium here and the gates that are involved here. So at rest, a neuron, the chemical gates are closed. We're going to slow this thing down like molasses. All right? I mean, we're going to just break this thing down step by step into, it's really not too bad, but we're going to break it down into step by step, and then you're going to be able to start rebuilding this story and telling it to yourself faster and faster. So at rest, the sodium gates are closed. That's good. I'll call them gates, channels, whatever. When the membrane is exposed to something, some sort of stimuli, if this was a neuron related to vision, it might be light. If this was a neuron related to taste, this might be a chemical. If this is a neuron related to hearing, this might be a sound wave. But something triggers and causes a disturbance in the membrane. That disturbance is going to open chemical channels. Okay, you tell me. I'm a neuron. I've been stimulated, and as a result, sodium gates open, right? Channels open. What's going to happen? Gate opens. Sodium, always found greater on the outside, comes flooding in. Yes? Can you picture it? What's happening now to the inside charge?
More positive.
Positive signals are coming in. The inside's always negative. And I've got more and more positives coming in. So now the inside is becoming more positive, aka less negative. This reduces the reduces the membrane potential. And the cell from the inside is going to go from negative 70. Remember I told you to memorize negative 70. And what's happening to this number? Less negative, the number is going up. We're going to say that the membrane is becoming depolarized. Key word, depolarized. What does that word mean to you? Can you picture, when I say polarity, what comes to mind?
Charge.
Charge, differences, right? There's a polarity across the membrane. There's more positive on the outside, more negative on the inside. If the inside is becoming less negative, that means that the difference between the inside and the outside is becoming less significant, isn't it? That means we are depolarizing, right? If there was a greater polarization, there would be a greater difference inside from outside. So we're going to use this word depolarizing to describe the charge moving upward, more positive. Wait till the graph comes in a moment. Okay, so there's been a disturbance. Sodium gates open. Sodium rushes in. We start this rush of charging going in. Now, the sodiums are being brought in by two there's two things driving the sodium in. What are the two things driving sodium into the cell?
Electrical.
Chemically, right? Chemical concentration, where is there more sodium? Outside. It wants to come in, right? Good old diffusion. But also electrically. Sodium is positive, the inside of the cell is negative, it wants to get there, right? So we've got two things pushing sodium into the cell very, very rapidly. A concentration and an electrical gradient. Fair enough? And that's going to cause the inside of the cell to become less negative. Okay, so the sodium's going to come rushing in, rushing in, rushing in, rushing in. This is still a local current. This is still a change in a very local environment. The whole cell's not becoming positive. Just that little area where the sodium gate opened, okay? I think one of the misconceptions is when we start reading through this thing, we say, oh, the sodium's rushing in. The entire cell on the inside becomes positive. No, just in that little local place. Okay, this is an analogy that just popped in my head, but imagine we had a gate, right, across a few miles, and if there was any break in that gate, right, there'd be a change in the concentration at that little place, but the entire gate would not have been flooded open, okay? So we're talking about little tiny localized changes. So in this picture here, again, what's happened? Sodium at rest, this is -70 in the fine print, and there's been a stimulation to the neuron, and as a result, sodium, see the sodium gates are open, sodium gates are coming in, and the inside there's -65, and eventually it just keeps getting more and more negative, right? So the local current is coming in, and it becomes, there we go, -60 even. So there's a slight change in the local current. This is still a graded potential. And how much effect this little bit of sodium coming in, you know, it's going to have a little bit of effect to the neighboring areas, but it's not going to have this massive change. So again, we still call it a graded potential, just a little bit. Now, what if we keep going now? What if we keep, the gate opens and more sodium comes in, such that, right, the disturbance becomes greater. And we keep opening more and more gates until the point where we have even more sodium coming in and even of a greater depolarization. The floodgate's really been opened, right? Kind of becomes overwhelming. So where this happens, look at this. This is the blow up of a neuron. And where are we, folks? What we're showing here, here's an electrical stimulation coming in at one dendrite, one area. Here's another coming in over here. Those are local potentials. Right? Two little disturbances coming in. And where are they heading? Physically, we see that these, right, what does this represent right here? This little guy that's sitting here attached. What does that represent, that thing right there? That's a synaptic knob coming in from another neuron, okay? And it's making contact with the soma right here in this area that's highlighted in red. Do you agree that in that local environment, right, the hot red area, there's going to be a lot of changes because a lot of sodium channels have just opened up. So in that localized area, there's a big effect. And if enough of these signals come in, then you start having a larger effect in the overall neuron. We're really close to that axon hillock. Okay. And you see that the membrane potential, right at rest, -70, and the more disturbance there is, the redder it is, the more that we're changing the membrane potential, and we see that it's drifting upward. Well, imagine we just keep doing that. We just keep doing that, and more sodium comes in. But once you remove that chemical stimulus, remember, what is it that drove the sodium channels open? We started this conversation about 6 slides ago saying that the sodium gates open in response to a chemical. If the chemical goes away, now the gate closes, right? And the membrane would return to a normal resting membrane potential. Excess sodium would be transported out of the cell. How? Right, sodium potassium pump will bring everything back to normal, get rid of that sodium that came crawling in, and just kind of get everything back to resting homeostasis. When things come back to normal, we're going to call that repolarizing. So we depolarize it a little bit, and then when we restore things back to their normal resting membrane potential, we're going to say that we have reestablished polarity, right? Repolarized. Reestablished the polarity. Question?
Yes. Do you have to wait for the removal of the chemical stimulus for the pumps to start working? So they all
No, that's a good point. So let's say there's a chemical disturbance on the neuron. As a result, sodium gates open, right? There'll be a local disturbance, and there'll be sodium rushing in at that little place. The sodium-potassium pump's running all the time anyway. It's in the background, okay? So it's not stopping. It just can't keep up. So for a moment, right, there's more sodium coming in than the sodium-potassium pump can maintain. And so overall, the cell becomes depolarized. And then once that chemical stimulation is removed, sodium stops flooding in. The sodium-potassium pump helps restore us back to a repolarized state. Now, something else happens though. In addition to sodium gates opening, it turns out that there can also be changes in the membrane potential from potassium gates opening. Now, what happens if there's a potassium gate that opens? Potassium wants to go where? It wants to get out of there, right? It's already in higher concentration on the inside. So if you open a potassium gate, that means the inside of the cell becomes more. Let's do some gymnastics here. If we're losing positive charges out of the cell, then the cell is going to become more negative. We're going to say now that the cell is becoming hyperpolarized. It's going even more negative than negative 70. So the cell becomes hyperpolarized, even more negative than it was. So if we open sodium channels, we're going to depolarize. If we open potassium channels, we're going to hyperpolarize. We've got to get these words down. So take a look at this graphical representation to put all this together. So this is our resting membrane potential for a neuron, negative 70 millivolts. Everything's fine. The straight across line is the resting membrane potential. There's a little bit of a disturbance, a chemical disturbance that drives some sodium channels open. What happens when sodium channels open? What happens to the millivolt reading that I've got this little millivolt, right? This little voltmeter, and I'm sticking the little probe inside the cell. If I open up the sodium channels, what happens to the reading on that voltmeter?
Goes more positive.
More positive or less negative, and I see that it's going to rise up. In this case, it looks like it's going to run minus 60, okay? Less negative, more positive. And then we close that chemical signal, and so the sodium channels close, and it, quote, repolarizes, right, as it comes back down. And then it sits there. Nothing's happening. It's a boring neuron. It's at rest. And then for some reason, there is a chemical stimulus that opens a potassium channel. It happens. Now, as we just described, potassium, as it leaves the inside of the cell even more negative. That downward reading on the voltmeter we would call a hyperpolarizing change. Then those potassium gates would close, and we're back to resting membrane potential again. This is what cells are doing, right? Little sodium, little potassium, depolarizing, going up, hyperpolarizing, going down. That's the deal with a graded potential. Graded potentials can go up or down. They can be positive, they can be negative. Graded potentials don't go very far. Okay? So looking at this little table from your book, membrane potential affected for a very short distance, just moves around locally, okay? And it can either be depolarizing or hyperpolarizing. That's a key for graded potentials. Remember, the other word for graded potential is a local potential. Okay, same thing. Local or graded potential. And another thing is that the stronger the stimulus, the greater the change in the membrane polarization. So if there's a lot of chemical, there'd be a greater or lesser amount of this curve. Okay, hold on to that thought as we compare now a local or graded potential with what will be referred to look at those review questions, as what's going to be referred to as an action potential. Compare and contrast here. Action potential is going to occur down an axon. An action potential will move all the way from the cell body all the way down to the axon terminals. And what's going to happen, what's going to cause an action potential to occur is a bunch of graded potentials. So these little local or graded potentials are all going to be coming into the soma. And if these signals are great enough, they're going to push, if you will, the neuron into, or convince it to do an action potential. Okay? So an action potential propagates all the way down to the end of the axon. Now, an action potential is going to require all these different channels to be working. And we'll see that there are going to be leak channels, okay? So leak channels, where did I describe leak channels? Resting membrane potential. There were leak channels, things just move on their own. There were chemically gated channels. And there are, in a moment, we're going to see action potentials, and we're going to see that action potentials depend upon voltage-gated channels. So hold on. Like I said, there's always a lot of details that need to come together in putting this story together. I'm going to break down the steps of an action potential step by step. Then we'll look at a figure of this, and we'll rejoice that we understand it. Okay. So at rest. Cells not stimulated. It's just sitting there. The voltage-gated channels, both sodium and potassium, are closed. Cells just hanging, doing nothing. Something happens. There is some stimulus. There's going to be a depolarization because that stimulus is going to open some sodium channels. And that's going to be enough to open up some of the sodium channels. And we're going to depolarize. And at some point, about negative 60 millivolts, something is going to start happening. We'll define it more in a moment. We're going to start reaching toward threshold, and we'll talk about that threshold in a moment. Now, as these sodium channels open, even more of them will open. It just kind of, one opens, and then they all start opening up. They kind of cause each other to open because these are voltage-gated channels. Why are they opening? In response to voltage changes, right? So as one sodium gate opens, voltage changes occur, and more sodium gates open because they are activated by electrical changes. So now you've got a huge rushing in of sodium. This is going to cause a very rapid membrane depolarization. And it's going to move from about a negative 60 up to a very positive value. Now, at some point, as this is occurring, these sodium channels are going to close, and some potassium channels, which are also going to be triggered by voltage change, are going to open. But we know what happens. When potassium channels open, things move in different directions, don't they? So this curve, we're going to see in a moment, is going to go all the way up to positive 30 millivolts. The sodium channels will close. The potassium channels will open. Potassium will now start doing what? Rushing out, leaving the inside more and more negative. And as it becomes more negative, we're going to start repolarizing the membrane, bringing it back to where it had been. The potassium channels will close, but there's always going to be a little bit of an overshoot. And we're going to see that not only is the potential going to go back down to negative 70, but it typically will stay open just a little bit longer, and that will cause a little bit of a downward dip called in hyperpolarization. So there'll be a little bit of an overshoot as a hyperpolarizing moment. Now let's take a look at this in a couple different ways. This is the cartoon representation. Let me break this down for you. So we've got, here we are, we're in the neuron. We're at the area called an axon hillock. There has already been lots of stimuli coming in. So these arrows coming in represent local potentials, graded potentials, little nudges, little stimuli. If all of these little local stimuli create enough of a change on the membrane potential at the axon hillock, then we will say that the neuron has reached its threshold. Once a neuron reaches threshold, it's like floodgates all open up, and all of a sudden, there's just a ton of sodium gates that are in this area in the axon hillock, and they're going to open up. Okay? And our book uses -60 as the threshold. So -70, resting, -60. Some books will say -55, somewhere in that range. There is a threshold. Once you hit -60, boom, you've hit threshold. All the gates along the axon hillock open up. Sodium comes rushing in, and the voltage goes all the way up to +30. So there's been this huge influx of sodium into the cell, making the inside positive compared to the outside. Once this huge influx of sodium happens, the sodium gates will close. The potassium gates are just a little bit slower to respond. So now the potassium gates will open. Well, we know that when potassium gates open, right, as potassium gates open, over here, the orange guy, potassium's leaving. That's going to start bringing the inside more negative. In fact, it's going to overshoot. It's going to correct it. It's going to overcorrect down below -70, down to about -90 or so, hyperpolarizing, and then it will eventually return back to resting membrane potential at -70. So let's put this into, an image that you're going to be able to draw and make good sense of, because this is going to be something you're going to know really, 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 really well. Let's take this, and I'm going to skip over a slide and then come back to it. So I skipped over one slide, and I'm putting all this together into one diagram. Here's what this is telling us, and it's all numbered for us. In this diagram, the cell is sitting at a resting membrane potential of -70. That's the solid line down here. If it gets up to a certain point, it gets up to threshold. The threshold is shown as a dotted line at about -60. As it gets up to -60, there is a threshold that's reached, and when the threshold occurs, there's a rapid opening of tons, that's a big scientific word, of sodium channels. And in comes rushing sodium, overwhelming amounts of sodium. That causes, in that little part of the neuron, a depolarization, and we see the swing go up to +30. That's as far as it will go. Then the potassium gates will open. They're just slower to respond. Sodium's closed, potassium's open. That's going to repolarize, bring the charge back down to -70. However, there's always an overshoot. And now we'll go down below -70, down to closer to -80, -90, somewhere in that range. That's what this downward curve represents. We saw that a few minutes ago when I talked about potassium gates opening, and we called that downward overshoot hyperpolarization. An action potential always looks like this. It doesn't change. This is the other thing. When I said local potentials, what did I say? They can be positive or negative, right? And they only go a short distance. Action potentials always look exactly the same. They always go up to +30. They always come back down and overshoot a little bit and come back to resting. So always positive, if you will, and they go a long distance. They don't just go for a short distance within the dendrite or the soma of the neuron. They travel down the entire axon. And that axon can be really, really long, right? And you, and me especially, axons can be a meter long, going from your spinal cord all the way down to your big toe. So you're going to have these signals traveling all the way down the axon. They're not going to be interrupted. They're going to go all the way down. And we'll talk about myelin and some other things that can go wrong and what contribute to that in a few minutes. This diagram also has two zones that are highlighted, one in orange, one in blue. Absolute refractory period versus the relative refractory period. Refractory period refers to the time when the neuron could not fire again. Now, if you look at the bottom, we're talking about milliseconds, right? Milliseconds, things that we have a hard time even imagining how fast that is. As soon as the sodium gates open and go up and the potassium gates open and come down, that orangey period during which time there's a rapid influx and efflux of ions, there is absolutely no way that that neuron can fire again. That is the absolute refractory period. Absolutely no way can another stimulation cause another action potential. So once the gates open, you can't retract it. It has to go forward. And there's a time, very short period of time, about a millisecond, during which no matter what you do, that action potential cannot, another action potential cannot fire. Versus the relative refractory period, notice it's in this zone a little bit further down. And in this period, the sodium and the potassium gates have already opened and closed. It would be more difficult, but not impossible, to fire another action potential during the relative refractory period. So going back, looking at those definitions on the slide before, an absolute refractory period, a time during which, no matter what, no matter how strong, the membrane cannot respond to another stimulus. It's in the middle of the gates opening and closing, and it cannot initiate another opportunity, another event. Relative refractory period during a time when, if the stimulus is strong enough, you could cause another action potential to jump, okay, to go. So this table, this chart, this is figure 11-10 in your textbook. This needs to become something that you know well. And on the first exam, I can assure you, you need to understand this graph and perhaps even be able to draw it out, okay? And so what do we have on here? The key words: -70, resting membrane potential, threshold, and then this upward swing, depolarizing, then a downward swing, repolarizing, and then the overshoot, hyperpolarizing. So label those three parts: depolarization, repolarization, and hyperpolarization. So let's take a look at this video and pull this all together. Looks like I'm having a sound issue. Hold on, let me see if I can get this fixed. We had sound on last Thursday, right? So we should be okay here. Come on. I can't even get that to pop up. Let's see if I can get some sound. If not, I'll be reading to you.
Neurons send signals over long distances by generating and propagating action potentials. Most action potentials originate near the axon hillock of the cell body in the initial segment of the axon. It then travels the entire length of the axon. A closer look reveals that during an action potential, voltage-gated channels open and close, altering the permeability of the plasma membrane to sodium and potassium ions. A threshold stimulus changes the shape of the voltage-gated sodium channels, causing their activation gates to open. This event marks the beginning of phase 1 of the action potential, known as depolarization. As sodium ions diffuse into the axon, the membrane potential becomes less negative. This causes more voltage-gated sodium channels to open, and the membrane potential soars to plus 30 millivolts. At this point, two key events occur. The inactivation gates of voltage-gated sodium channels close, and voltage-gated potassium channels open. These two events mark the beginning of phase 2 of the action potential, known as repolarization. As potassium ions diffuse out of the axon, the membrane potential becomes negative again. However, the membrane potential continues in the negative direction, going beyond the resting state of minus 70 millivolts. This marks the beginning of phase 3 of the action potential, known as hyperpolarization. During this phase, voltage-gated potassium channels close and all voltage-gated sodium channels are released from inactivation. By the end of this phase, ions move through leak channels only and the membrane potential returns to the resting state of minus 70 millivolts. The neuron is now ready to fire another action potential. Summary. Generation of an action potential. A threshold stimulus opens voltage-gated sodium channels. Sodium ions diffuse into the axon, depolarizing it to plus 30 millivolts. Voltage-gated sodium channels close, and voltage-gated potassium channels open. Potassium ions diffuse out of the axon, repolarizing it to a negative value. The membrane potential briefly hyperpolarizes. Voltage-gated potassium channels close, and the membrane returns to the resting state of minus 70 millivolts.
Know it, right? Know this little story and know it well. And then as you're thinking about this, compare and contrast action potentials with local potentials, okay? And here's one way of thinking about these two, comparing them one to another. Graded potentials, the pressure on the trigger of a gun, right? So a little bit of pressure, a little bit of pressure, a little bit of pressure, but at some point you reach a, quote, threshold. Boom. You pull that now, and there's no going back. The gun is going to fire. So that's kind of the difference between a graded potential, aka local potential, and an action potential. With enough stimulus, with enough pressure on the trigger, then you will reach threshold and it will fire. Okay. Now there's a couple different ways that you could do that. What if I grabbed a gun and I pulled on the trigger just a little bit, and then a minute later pulled it again, and a minute later pulled it again, just a little bit? Am I going to reach threshold?
Nope.
Just a little bit, just a little bit, just a little bit. So if I spread it out over a long period of time, I'm not going to reach threshold. What if I squeeze just a little bit over on one side and then squeeze? Imagine I've got the trigger and I can cock it one way. So I'm pushing on this side, right? But I've also got other pressures going on the other side. So if I was pushing these things back and forth, and they're just kind of moving back and forth, I wouldn't blow the trigger either, would I? So these forces coming in, these local potentials, have to be coming in rapidly enough, right, to push it to threshold, and they all have to be telling the cell to do the same thing. So if I was pushing in one direction and pushing in the other direction, in other words, do it, don't do it, and if these two things were coming in, basically the trigger wouldn't go. So we'll talk about this idea of summation, summing up all the signals so that the neuron reaches threshold. And the other thing about action potential, I mentioned it, it's an all or none. When you hit it, it's going to go no matter what. It's going to reach the end of the axon, and that is quite different from a local potential. Okay, so it's an all or none. Either you reach the trigger point or you're not. Either you pull the trigger hard enough or you don't. And that's why sometimes the axon hillock is called the trigger zone. Okay, so you'll hear about in axons, the axon hillock called the initial segment of the axon, but you'll also hear it referred to as the trigger zone, because if there's enough stimulus that it reaches threshold, then the gun fires and the bullet will go down the entire length. The action potential will go down the entire length of the axon. Okay. Thoughts, questions, concerns. Crazy stuff here. Yes, sir.
Are we going to have to know about the sodium inactivation gate? I think it was in the video, not seeing the notes.
What happens is, okay, so as you're going back to the curve, tell me what's going on right here. What's opening at the arrow? At that arrow, what's opening? Sodium. Why did the sodium gates open? We reached a certain electrical threshold. Therefore, those are voltage gated, right? They open in response to a voltage change. So those are voltage gated sodium channels. They open at around -55. They opened up. Sodium went flooding in, and there's a depolarizing. But what happens here at the very top of this curve? The video told us, and I've mentioned, here the sodium channels were, quote, inactivated. They close. Okay, so they just kind of close at this point. But the sodium gates are going to close, but the voltage gates are now going to open. Okay, so the activation of the gate opens it, the inactivation closes it. In this case, it's all related to voltage. So voltage-gated sodium channels open, then close. Potassium gates will open at the top and then close as they come down toward the bottom.
At that same point.
Pretty much that same point. So there's kind of this two important things happen at the top of the hill there, right? So you've got the sodium channels closing, the potassium gates opening. So now you imagine what happens, right? Now potassium is going to be, as we've said, rushing out, making the inside of the cell more negative, and we see the voltmeter bringing the number back down, repolarizing the membrane. It takes a little while to get this to sink in, I know, but I know that it will. So that's 11-10. Now, just a few more things. How does this action potential go? We're describing right now at the ionic level, at the sodium and potassium level, but there's a few more things I want you to know about action potentials as they go down in axon. Number one, there's two different kinds of propagation, two different ways. Once the trigger is pulled, once the action potential travels down the axon, there's two basic patterns of movement. One, continuous. Two, saltatory. Continuous propagation is what happens when there is no myelin. Okay, so right next to continuous, write unmyelinated. Next to the word saltatory, write the word myelinated. Okay, so continuous, unmyelinated, it'll be on the next slide. Saltatory is myelinated. Anyone recall what myelin is?
It helps it's like an insulation.
It's like an insulation blanket around only the axon. It's a waxy, white in color, lipoprotein, so it's kind of fatty protein. It is made by what kind of cells?
Glial.
Glial cells means helper cells in the peripheral nervous system. What cells make the myelin? Schwann cells. If I had said we're in the central nervous system, oligodendrocytes are going to make the Schwann cells, okay? Just a little reminder, some anatomy things. Now, not all axons, though, are equally myelinated. Some axons are said to be unmyelinated. That means that the myelin is not wrapped around it in the same way, leaving a portion of the axon exposed, not completely wrapped. As a result of that, it's going to take a lot longer for this sodium-potassium change to occur. We're going to call this continuous conduction. It's slower. 1 meter per second. That's pretty fast, right? 1 meter per second, but we're going to see how slow that is for this kind of signal. So continuous propagation, unmyelinated, longer, 1 meter per second or thereabouts. And here's what's going on. As this action potential is going down the axon, there are still sodium-potassium gates opening and closing down along the entire length of the axon. So it just takes a long time for that to occur if this thing is not wrapped in myelin. what we're going to see in a moment is that if there's myelin wrapped around the axon, it's almost like the action potential can skip jump, right? Almost like taking a little rock and slinging it into a pond, and you see it, right, skip jump over areas. And that's what happens when there's myelin. But when there isn't myelin, this progression of steps, you can read through this, but basically that little sodium, potassium, -70, -55, -70, -55, that has to occur down the length of the axon. And that's just a long, tedious process versus saltatory conduction. It's a weird word, but I remember that saltatory, when I hear the word salt, I think of sodium chloride. Has nothing to do with saltaire, means to leap, okay? But saltatory, I just think, you know what? I know that an action potential has a lot of sodium, potassium going on, and when there's myelin around, that signal can jump, that salt bridge, if you can jump from place to place. If the myelin is all the way around the axon, then you know there's a space in between the Schwann cells, or in between the oligodendrocytes, called a node. Node of Ranvier, right, is the space between the Schwann cells. The area that is myelinated, we would call the internodal region. Let's make sure we're okay with that. That was in your lab last week in one of those questions. But this word internode. So if this is a Schwann cell, and this is a Schwann cell, and going down the center of it is the axon, right? And there's another Schwann cell. So what are these areas here called? The arrows represent nodes of Ranvier, right? In between. What would we call the area that is myelinated, that is covered by the Schwann cell? Internodal. Think between the nodes. So in between the nodes, there's an area where the Schwann cell resides, where it is completely myelinated. It turns out that that myelin is going to allow the signal to travel faster. It does insulate and protect the axon, but it also speeds up the conduction. So now basically the action potential, it'll start down at the initial segment, think axon hillock, trigger zone. It'll start there, and it will travel by skipping. It skips over the internodes, right? It skips over the internodes and goes from node to node to node. Skip jumps all the way down. And this is much, much faster than continuous conduction. Another thing that's going to contribute to how fast this can occur, the larger the diameter of the axon. When we look at histology of neurons, we're going to see that not every axon is the same diameter. The bigger diameter, the more surface area there is. The more surface area there is on an axon, the more sodium, potassium channels there can be, and the more rapidly the conduction can occur. So bigger diameter, faster conduction. Myelin, faster conduction. Okay? So the bigger diameter, faster conduction, less resistance to ion movement, a number of different ways to think about why this occurs. So what we see now is that in this diagram from your textbook, you'll see the description where the signal is going to travel from node to node to node, and where there's a Schwann cell, Schwann cell, Schwann cell, it basically can jump over that area, jump over the internodal regions to the nodes, and propagate much more quickly. So let's take a look at this little video and make sure we got this story down based upon this description.
Action potentials propagate in a continuous fashion in unmyelinated axons. Once an action potential is generated in the initial segment of the axon, it propagates the entire length of the axon. Recall that a threshold stimulus causes voltage-gated sodium channels to open. The influx of sodium ions generates an action potential. It also establishes a depolarizing current that flows to the next segment and brings it to threshold. Voltage-gated sodium channels open, regenerating the action potential in this segment of the axon. Current flows from this segment and depolarizes the next segment to threshold, thus regenerating the action potential yet again. In this way, regeneration continues in one direction all the way down to the axon terminals. the basis for unidirectional propagation is revealed when we take a closer look. By the end of the depolarization phase of the action potential, all voltage-gated sodium channels inactivate and voltage-gated potassium channels open. These two events render this segment of the axon temporarily insensitive or refractory to another depolarizing stimulus. However, voltage-gated sodium channels in the downstream segment are closed and receptive to a depolarizing stimulus. Thus, propagation occurs sequentially down the axon to the axon terminals. In myelinated axons, action potential propagation is a bit different. Here they propagate in a saltatory or leaping fashion. The myelin sheath consists of multiple layers of tightly wrapped glial cell membrane. But this sheath is not a continuous one. Exposed areas of axonal membrane, known as nodes of Ranvier, occur at discrete intervals. Voltage-gated sodium channels are abundant in the nodes, but largely absent between nodes. So, action potentials are regenerated at each node, not in areas covered by the myelin sheath. However, the myelin sheath does provide the insulation necessary for the rapid spread of depolarizing current. And the sooner the nodes reach threshold, the faster action potentials propagate along the axon. Saltatory conduction is extremely fast. Velocities often exceed 100 meters per second. In contrast, continuous conduction is fairly slow. Velocities rarely exceed 2 meters per second. Nevertheless, both continuous and saltatory conduction propagate action potentials over varying distances because action potentials regenerate along the way. Summary: propagation of an action potential. Once generated, the action potential propagates the entire length of the axon without decrement.
See how the curves are all the same? Every action potential is exactly the same. Negative 70 up to positive 30, negative 70 up to positive 30. So always exactly the same visual look to every action potential curve. I don't think I didn't publish it here, but she was saying that saltatory conduction is 50 to 100 times faster, isn't it? 50 to 100 times faster than continuous propagation. So amazingly fast speeds as signals are traveling through our body and connecting one neuron to the next. Okay, so thoughts, questions, concerns over action potentials. It's kind of tedious. I think if you'll interact with the mastering content, you'll be asked lots of questions attacking or looking at action potentials from many different ways. What's happening to the membrane? What's happening to the receptors? What's happening to the gates? What's happening to the sodium, the potassium? What's happening to the overall voltage? And if you entertain this from enough different angles, then you will walk away understanding the big picture. Yes, sir.
The Chapter 11 masterings that are for tomorrow.
Yes. There's a couple different kinds of mastering things on there. There's one that's the content questions to kind of start digging into it. The homework won't be due until later. Okay, but this is a content-specific little thing. Yeah. So right now the deadlines are good in mastering, and I want some feedback. Okay, so some nice constructive feedback about the mastering assignments, and I'll make adjustments as necessary. I've got quite a few on there right now. Right, you're kind of busy. You've got something going on every day or two. Take a look at that. Try not to miss any deadlines. Work ahead. Some people have already told me they've worked ahead through pretty much everything that's posted. They're done for the month. So just take a look at it and try not to miss too many assignments. I will hold the deadline strong. So I always believe that if 95% of the class could get it done, and you come to me with it may be a valid reason why you didn't get it done. But if I give you an extension, it's not fair to the people who stayed up to 3 o'clock in the morning to get the other things done by the deadline. So I'm going to hold you to the deadlines unless there's some catastrophic thing that occurs. So what happens when this action potential travels down the axon? What happens at the other end? Kind of breaking this down. what happens down at the axon terminal, where the axon swells a little bit at what we call the synaptic knob. That's where the neuron is going to come in close proximity with another cell. It could be another neuron, or it could be a muscle cell. It could be a gland cell telling it to secrete. Right? Something's at the end. That's what the whole purpose of this, right? Is to send a signal to affect change. So what's going to happen is that this information traveling down the axon is going to head down toward the synapse. And here, we're going to talk about pre- and postsynaptic neurons. So for now, we're going to describe the next cell as another neuron. All right, so forget muscle for a moment, forget gland for a moment. We're talking about one neuron is going to speak to the next neuron in the sequence. We're going to have a pre- and a postsynaptic cell. The synapse is the space in between. Two types of synapses. This should be somewhat familiar. Chemical synapses and electrical synapses. The idea here is this. Chemical synapses speak using chemicals. There's an electrical signal that travels down the axon. That electrical signal is going to cause the release of a chemical. Those chemicals are called collectively neurotransmitters. And then that neurotransmitter is going to tickle, bind to, touch the next neuron and cause another electrical signal. So we're going to go from electrical signal to chemical signal to electrical signal. That's the essence of a chemical synapse. Acetylcholine, right, the one and only neurotransmitter that I've discussed so far, is a commonly used neurotransmitter. We call it a cholinergic receptor. It has the word choline in it, cholinergic, so that's not hard to remember. We'll talk more about that later. There are also, in a chemical synapse, what happens is that a signal travels down. It's going to cause voltage-gated calcium channels to open up. Again, we have an electrical change. That electrical current reaches the axon terminus down at the synaptic knob. That voltage difference is going to cause calcium gates to open. Well, it turns out that calcium is found outside of the neuron. This is going to cause calcium to come rushing into the terminus. When that calcium comes into the terminus, it is going to cause and trigger exocytosis. Exocytosis, it's when little vesicles inside the cell are going to bump up against the membrane, open up, and release the contents. The vesicles, in this case, are going to be filled with this neurotransmitter called acetylcholine. So the electrical signal comes down, causes the calcium to come in, bind to the vesicles, cause exocytosis. Once that signal is gone, that voltage signal is gone, calcium goes away, and the neurotransmitters are no longer going to be released. We'll show a picture of this in a moment. That acetylcholine is then going to be released across the synapse, across what's called the synaptic cleft. And in a very short distance, there's going to be another neuron there. That neuron is going to have a cell membrane, and in that membrane, there are going to be ligand-gated channels, right? And that acetylcholine, ACH, is going to diffuse across and bind to a chemically gated sodium channel. What's going to cause this sodium gate to open? The binding of acetylcholine. So acetylcholine comes across, it's a chemical or ligand-gated channel. It happens to be a sodium channel. Surprise, surprise. So when acetylcholine comes across the cleft, opens up the sodium channels, and what now happens in the next neuron?
Graded potential.
A graded potential. Good catch, right? Sodium gates are open. Sodium goes rushing into the cell. But this is going to happen near the dendrite or near the soma, isn't it? That's not an action potential, is it? The action potential only occurs if there's enough stimulation to reach threshold at the trigger zone. So when that sodium gate opens, it's going to open another local or graded potential. Again, if there's enough of those stimuli in this postsynaptic cell to reach threshold, then and only then would you trigger another action potential. Now that's a chemical synapse. And again, what happens at the postsynaptic cell is temporary because this is a local potential. Now, there's an enzyme involved in a chemical synapse that I want you to know about called acetylcholine esterase. Okay? Acetylcholine is a chemical. If acetylcholine stuck around too long, then the next cell would be opening the sodium channels too long. So acetylcholine esterase comes along, ACHE, comes along and basically is like an eraser. And it's going to break down and destroy the acetylcholine. And it does so amazingly fast, 20 milliseconds or so, and it can basically clean up the entire cleft. That means that another signal could come down, more acetylcholine can be released, and we can have very, very rapid action potentials occurring and signals being sent between neurons. So here's the picture of all this. We've got our presynaptic neuron. Action potentials coming down as the arrow comes down. As it comes down, okay, there is going to be calcium that comes floating in. The reason calcium comes in is because this voltage coming down to the axon terminus is going to open up these voltage-gated calcium channels. They're shown here in the membrane. Calcium comes in. Calcium binds to these vesicles, which already have within them acetylcholine. Acetylcholine is a protein. Where was this acetylcholine made? This is amazing. Where was that acetylcholine made? Where do proteins get made in a cell?
Mitochondria.
Where do proteins get made? On the ribosomes, right?
Yep.
Back up in the soma. So if we're talking about the neuron in my big toe, that means that the soma for those neurons are up in my spinal cord. So the acetylcholine was made up on the ribosomes and up by the Golgi apparatus and the endoplasmic reticulum and all those good things that you've forgotten about. And the proteins are made stuck in those little vesicles by the Golgi apparatus. Remember, the Golgi is kind of the UPS center, sends out packages of things. And those little vesicles are going to travel all the way down the axon. Okay? This is a long trip. And then those vesicles are going to be sitting at the very, very end of the axon terminus waiting to be released. They're released, why? Because a signal comes down, calcium comes in, calcium binding to the vesicles causes exocytosis. Now, in this diagram, all these little red molecules are the acetylcholine. Acetylcholine now is going to bind to these receptors. On this membrane, those receptors are what type? Acetylcholine is a chemical, right? So these are going to be a chemical or a ligand-gated channel. They turn out to be sodium channels. So when the acetylcholine binds, it opens up a sodium channel. Sodium comes rushing in. The little half moon guys in here, little yellow guys, represent the acetylcholine esterase, and that's going to quickly wipe the slate clean, getting rid of any acetylcholine, bringing us back pretty much to rest. But what happened when that sodium came rushing in? This little arrow reminds us that we've now propagated a local potential, which could potentially become an action potential in the postsynaptic cell. So really, really cool stuff that happens. Here's a blow-up of that, easier to see. I'll let you look through the steps of this. Okay, 1, 2, 3, 4, 5. I think if you'll walk through this, you'll be in good shape. This is another thing to think about is synaptic fatigue. What if you were sending constant signals down to the end of the axon? At some point, it's possible that the neurotransmitters run out. Right? There's only so many neurotransmitters that can be sitting there waiting. So it's possible, not that common, but possible for either all of the acetylcholines to have been used up, or maybe they weren't produced enough in the first place, or maybe for some reason they're just not keeping up with the function. So there can be synaptic fatigue, where the neuron is just not keeping up with the neurotransmitters necessary. There can also be delay. How long does it take for the chemical to go across the synaptic vesicle, or sorry, the synaptic cleft? Typically 0.2 to 0.5 milliseconds, but if this gets longer for different reasons, then there could be a delay, a slowing down of signals talking to each other, and this is where a lot of our antidepressant medications work. They actually help to either speed up or to at least slow down the degradation of neurotransmitters. So a lot of serotonin, uptake inhibitor, those kinds of antidepressants basically cause the neurotransmitters to stay in the cleft longer, giving them a greater opportunity to affect change on the postsynaptic cell. So that's sort of where all those neurotransmitters are, you hear about them in every language with antidepressants. Then there are, I'll finish up with electrical synapses. This is where one neuron directly speaks to another cell, not through a chemical intermediate, but there's electrical signals that travel from one cell directly into the next cell. No neurotransmitters. Instead, there is what's called a gap junction. The cells, the pre- and postsynaptic cells, are linked physically together, and the electrical charges flow from one cell directly into the next. I've talked about these a little bit before. This is basically what intercalated discs are, right? So within the heart, within cardiac muscle, those intercalated discs, what we're seeing under the microscope really are gap junctions, and they are evidence of electrical synapses where signals are sent quickly from one cell to the next. We don't have to wait for chemicals. This whole chemical thing definitely slows down the propagation of the signal, but the one thing that these chemicals do is add to the complexity. So while you may be thinking, well, why in the world does the nervous system deal with electrical signal really fast, and then slowing it down for a chemical, and then speeding along again for another electrical signal? If our nervous system was all just zinging electrical signals, we wouldn't have much control. So these, as we're going to see, these synaptic clefts where the chemicals are, very complex, giving tremendous opportunity for our body to tweak, increase, decrease, interact, make different changes at these chemical synapses. And so we have a lot more control because of the chemical synapses. Here's a picture of an electrical synapse. And again, you don't see any chemicals. And this close proximity here represents direct touching of the pre- and the postsynaptic cell, and the electrical signal goes straight through, right? No chemical intermediate in an electrical. Think intercalated discs in cardiac muscle. There's also some of that going on in smooth muscle that we'll get to when we get to the digestive system. The last little bit of this is on EPSPs and IPSPs. EPSPs, read through this. It's what we've already described. These are basically what happens on the next cell. So when the acetylcholine is released, and what's it going to do to the next cell in sequence? Is it going to be excitatory, EPSP, excitatory postsynaptic potential, basically a graded potential that's excitatory, or is the signal going to be inhibitory? Remember I told you that local potentials could be either excitatory or inhibitory, up or down. Okay? That's what another way of saying these. These are local potentials, EPSPs and IPSPs, and what is an EPSP going to do? Let's think about this term together. If it's going to be excitatory, do you agree it's going to push the cell toward depolarization? It's going to shift it up toward threshold. If the next signal along is inhibitory, then it's going to cause the cell to become hyperpolarized. If it becomes hyperpolarized, it's going to do what? Make it less likely, more difficult for the cell to reach threshold. So if you're driving, remember I said the idea that a trigger, if there was such a thing as a trigger that you could push on both sides, right? And if I'm pushing on one side excitatory, but I'm also pushing with the same amount of pressure on the other side inhibitory, then the trigger is not going to move at all, is it? So if I have these incoming signals, some of which are pushing positively and some of which are pushing negatively, then they're basically going to cancel each other out. These EPSPs and IPSPs will negate each other, and they won't allow the next cell to go to threshold. So we'll talk about this idea of summation. Boy, I'm almost done with this chapter. We'll get through IPSP's summation, temporal and spatial summation. Please look at that. There is, if you've looked ahead, the next, the first exam is going to cover the stuff on water and electrolytes. Chapter 11, action potentials. Chapter, what's the next one? 13? Okay, and then chapter 15, and somehow 15 didn't make it into your course pack. So I am making copies for you, and on Thursday I will be handing you, for some reason, 10 pages got left out of your course pack, and you have nothing for chapter 15. So I'll be handing that to you live. I've posted it to Blackboard, but I will give you a physical copy, and you can slide it into your course pack on Thursday. So come see me if you have any concerns at all, and I will see all of you Thursday, if not sooner.